Hi, I am Amrita, Assistant Professor Krishna Jayanti University, and I want to discuss a molecular cell biology topic, which is biological membranes. So I want to start with explaining the structure and function of biological membranes. So every cell is surrounded by a bilayer membrane, and which plays a very important role, not just for protection, for defense against external agents, but it also has sensory perception as well as it has the property of signal transduction. So this is the structure of the membrane. As we can see, it is not a very simple structure. It is quite complex with the presence of intermembrane proteins, lipid bilayer with the lines that are shown, these are the non-polar regions, and the circles that are shown, these are the polar groups. So we have a bimembrane structure with polar as well as antipolar regions. Then there are peripheral proteins. Then there are these are the glycolipids. Glycolipids come from the term glyco and lipid. So in the lipid, there is a region, glucose region, sugar region. That's why the term glyco, similarly glycoprotein. And these are the structures that have the role in sensory perception, signal transduction, etc. And the membrane key components, if we see, it's the lipids that I showed, which has both the polar and non-polar ends. And these lipids have a particular role because the non-polar hydrophobic tails can come together to form a very compact structure, and the polar regions will be outside, and it has the capability, because of its structural components, it has the capability to form a compact region, and with all the molecules hanging to it, and looks like a raft because it's the lipid with certain specialized structures like glycolipids, proteins inserted in it, forming into a structure, something like a mosaic, and hence the structure is also called as a fluid mosaic model. And if we see here, certain regions are there where a lot of other components are present, like lipid components, sugar components, protein components. Hence, these are called as rafts. And there are also non-lipid raft regions where these components are not so concentrated. These proteins have a very important role that are present in the membranes. They are involved in the transport of certain components inside and outside, and not just transport and the cargo transduction, but also they are involved in certain functions like transporters, anchors, integrating through these components. There can be attachment of these cells to other matrix regions, and also through these proteins there are certain receptors that are attached to the membrane that gets involved in the ligand attachment and the signal transduction. Also, certain proteins have catalytic ability and are able to catalyze certain substrates into products. Hence, these proteins also have certain enzymatic functions. And lastly, it is the oligosaccharides that form the glyco part of the glycoprotein and the glycolipids, and they are uh, quite variable, and because of their variability they are also involved in attachment, communication, as well as adhesion, as well as signal transduction. The asymmetry is an important aspect, as we saw that certain lipid raft region had all the concentration of components, whereas other regions did not have those aggregation of components. Because of this asymmetry, there are certain regions that are more active for cell signaling, others that are not so active for cell signaling. So overall, this is the structure of the membrane that also explains the reasons for its variable function and differential properties. Thank you very much.